Unit 6 Reproduction. Today I will be doing sexual reproduction in flowering plants. Flowering plants meaning angiosperms. The topics in this chapter are flower, a fascinating organ of angiosperms, pre-fertilization events, structures and events, double fertilization, which is triple fertilization, triple fusion and syngamy, post-fertilization events, structures and events, apomixis and polyembryony. Let's continue reading the NCERT. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at diversity of structures of inflorescences, flowers, and floral parts shows an amazing range of adaptations to ensure the formation of end products of sexual reproduction. The fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants, that is, angiosperms. Whatever we see in the plants, the flower shape, the petals, the fragrance, all is not for our own selfishness, but it is a reproductive organ helping the plant reproduce sexually. And the end products of reproduction are nothing but the fruits and seeds we see. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relation with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers. Okay. This is the diagram of the flower. This is the basic diagram which I have been seeing from early standards itself. This is a diagram of the hibiscus flower or China rose flower. This is the diagrammatic representation of a LS, lateral section of a flower, where this is the thalamus, ovary, the stalk of the flower, which we call pedicel. If there is a stalk in the flower, we call it as pedicellate flowers. If there isn't, we call it as non-pedicellate flowers, and these are sepals. The stamen, the whole here is stamen, this being filament, and this part bilobed being anther. These are the petals, and the main female reproductory organ, which is what we call either pistil or carpel. To a biologist, flowers are morphological and embryological marvels and sites of sexual reproduction. In earlier classes, you have read that various parts of a flower. This figure will help you recall the parts of a typical flower. Can you name the two parts of flower which the two most important units of sexual reproduction develop? The flower is divided into four whorls of the two essential whorls and two non-essential whorls. The essential being the androecium and the gynoecium, which are nothing but the male reproductive part and female reproductive part, and the non-essential or accessory whorls being calyx, the first whorl, and the second corolla. Corolla are the petals, whereas calyx are the sepals. And the third whorl is the androecium, and the fourth whorl, third is the androecium, and fourth is the gynoecium. The whorl it is represented. That is the outermost. First calyx, second corolla, third androecium, and inside fourth is the gynoecium. Now let's start with the first topic. The first main topic, pre-fertilization events and structures. Much before the actual flower is seen in a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to differentiation and further development of the floral primordium. Let's read it again. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Floral primordium is from where the flower will develop and where there are hormonal changes. It is differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, androecium and gynoecium, differentiate and develop. You would recollect the androecium consists of a whorl of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. The first topic here in the pre-fertilization events: stamen, microsporangium and pollen grain. Stamen is the main male reproductive organ. Microsporangium is what pollen grain comes out of from. Microsporangium is MMC or PMC, and pollen grain is the final gamete. This is the representation. This is this whole is what we call a stamen. This part, this stalk, we call it as filament, and this part, which is bilobed, we call it as anther. This is filament, the stalk, and this is a section of the anther, where these are the tetrad structure, bilobed, dithecous, with the separation, connective tissue. This is the tetrad structure. A butterfly-like structure here. These figures show the two parts of a typical stamen, the long and slender stalk called the filament, and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The proximal end, which is this is the in an anther. If this is an anther, this is the anther. This is the stamen. If we read again, we will see that the long and slender stalk is called the filament. The terminal and generally bilobed structure is called anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. This is a stamen, and this is the anther. This is the stamen, this is the anther, this is the filament. This is the proximal end of the filament, proximal end, whereas this is the distant end. This proximal end is what we see is attached to either thalamus or petal of the flower when the condition is epiphyllous. And remember that this part is what we call the proximal end of the filament, and automatically this part will become the distant end. Proximal, distal, convoluted tubules, we remember as such here as well. This is the distant end, and whereas this part is the proximal end. Continue reading. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range and shape in attachment of anthers in the flowers. We see many conditions like monoadelphous, diadelphous, where the stamen can be single or bundled, or in lily we see that there is a condition where in the lily flower it is the epiphyllous condition where stamen are attached to the petals. In various different kind of species we see differences in in stamen. We see differences in different kind of flower. We see differences in number of stamen, arrangement of stamen, third, sizes or length of stamen. We see that in some flowers, 2 stamens or 4 stamens will be long, whereas 2 other stamens will be small. This kind of arrangement, variation in sizes of stamen, sizes or length of stamen, and they can be either free or fused, and there can be 2 bundles, 3 bundles based on number of bundles as well. As such, this is the variation we see in stamen. A typical angiosperm anther is bilobed with each lobe having 2 theca, that is, they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Generally, most of the angiospermic anther are bilobed, meaning this is the stamen, this will be the anther. If we take a sectional view, see that this is the horizontal view. These are the 2 lobes. Each lobe is dithecous. If this is one lobe, each lobe is dithecous. It is divided into 2 theca. So the total anther, a bilobed anther, contains 4 theca, but each lobe contains 2 theca, which is why we call it as dithecous. Theca nothing but separation. This is a longitudinal groove they mentioned, that is this one, that is separating the theca. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of an anther. This anther is a 4-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. He is calling microsporangia. These ones these are nothing but the microsporangia. The microsporangia develop further and become pollen sac. They are extended longitudinally all through the length of an anther and are packed with pollen grains. Here we see the description of that. Here if you see, these are the, and till as this is the length of the anther, till here there will be 2 pollen sacs, which we see as here contain pollen grains, like here, pollen grain. We should remember that microsporangia, there are 4 microsporangia at the 4 corners of a transverse section of an anther. This microsporangia develop into pollen sacs. And now next, structure of microsporangium. In a transverse section, a typical microsporangia appears near circular in outline. It is generally surrounded by 4 wall layers: the epidermis, endothecium, middle layers, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of the anther to release the pollen grain. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possess dense cytoplasm and are generally have more than one nucleus. Can you think how tapetal cells could become dinucleate? When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. Let's read again. In a transverse section, a typical microsporangium appears near circular in outline, is generally surrounded by four wall layers: the epidermis, endothelium, middle layers, and tapetum. The outer three wall layers perform the function and protection and help in the dehiscence of release of anther to release the pollen. The innermost wall layer, tapetum, it nourishes the developing pollen grains. Cells of tapetum possess dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupy the center of each microsporangium. This is a diagram of the transverse section of the young anther. This is the structure where the outermost is the epidermis. This part, epidermis. The next part is the endothelium. The next being middle layers. The next, these are the compactly arranged homogeneous tissue, which we call sporogenous tissue. And these cells are the tapetal cells. And this is the connective tissue. Basic diagram. And the first three layers of epidermis, endothelium, middle layers help their functions. These three layers functions are divided into one: protection of the anther, two, help in dehiscence of anther, dehiscence meaning to help in releasing or opening of the pollen, opening of the anther to release the pollen grains, dehiscence of the anther. These are the two functions of the first three layers of the epidermis, endothelium, and middle layers. As for the fourth layer, which is tapetum, the fourth layer, tapetum, these features are: one, dense cytoplasm, binucleate or possess many nucleus, their function being nourish the growing pollen grains, These are the three functions of the fourth layer, whereas the first two layers possess only two functions, which is protection and help in dehiscence of anther. This is the cross-sectional view. Increased magnified view here. This being epidermis, endothelium, middle layers, tapetum cells, and the microspore mother cell or pollen mother cell. Transverse section of a young anther. This is another This is the measure dehiscence anther, where we see that there is no separation here and it got opened. For dehiscence, these pollen grains will come out, which may be through anemophily, maybe entomophily. They will get pollinated later. Pollen out. Let's continue reading. As each cell of sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells. The microspore tetrad as the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspore or pollen grains are formed that are released with the dehiscence of anther. Basically, there will be in the sporogenous tissue here, this is the sporogenous tissue. In this sporogenous tissue, which are compactly and homogeneously arranged, there are pollen mother cells which are diploid and which will undergo meiosis 1 and meiosis 2 to produce 4 pollen grains. But in the earlier stages, in the young stages, this 4 pollen grains, even after division, will remain stuck together, which is called microspore tetrad. The cluster of 4 cells, because one PMC, as it is 2N, when it undergoes meiosis, it will produce 4 cells, 4 haploid gametes, and these gametes being stuck together as a cluster of cells, a cluster of 4 cells, which is what we call a microspore tetrad. This microspore tetrad, as the anther matures and dehydrates, this microspore tetrad will also be separated to form individual pollen grains. So we can say that each PMC or pollen mother cell can produce 4 individual pollen grains or microspores. Now let's go to pollen grain itself, which is another name of pollen grain being microspore. Pollen and for ovule, if we have another name of embryo sac is megaspore. Ovule is megasporangium. Here in the male reproductive or andricium, it's better to write andricium. In andricium, the microsporangium is the PMC or pollen mother cell, whereas the microspore or male gamete is the pollen grain. In gynoecium, the microsporangium is ovule, whereas the This is not the microspore. This is the megasporangium is the ovule, whereas the megaspore is the embryo sac. In androecium, microsporangium is the pollen mother cell and microspore is the pollen grain. In the gynoecium, the megasporangium is ovule and megaspore is embryo sac. Just read that. Let's see the pollen grain. Pollen grains represent the male gametophytes or gametes. If you touch the open anthers of hibiscus, china rose or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains. A few textbook pictures they gave here is yes, and this is the microspore tetrad. Microspore tetrad, as during the young stages of the anther, the microspores after the complete division, after the division of the pollen mother cell, will be stuck together as a cluster of 4 cells. Here we see 4 nucleus, and this is what we call a microspore tetrad, a cluster of 4 cells. This is a diagram, and these are the types of pollen grains formed. It can be spiky like this, spikes. It can be in variety of shapes like this, small cactus like, or many shapes depending on the species of the plant. And as we see in the later of the chapter, we know in aquatic plants the pollen grain, where in aquatic plants, if they undergo hydrophily or pollination via water, in that kind of plant we see that the pollen grain is ribbon shaped or helix shaped, and it is covered by a gelatinous layer. This is also one of the different adaptation of pollen grain. So let's continue. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. 25 to micro meaning 10 power minus 6, 25 to 50, 10 power minus 6 meter or micrometer, which is very small, 25 to 50 micron meter in diameter. This is the pollen grain. If we assume the diameter will be 50 to 25 to 50 micrometer. It has a prominent two-layered wall, two walls. The hard outer layer called exine is made up of sporopollen, which is one of the most resistant organic materials known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollen is so far known. The pollen grain exine has a prominent aperture called germ pores, where sporopollen is absent. The pollen grains are well preserved as fossils because of the presence of sporopollen. The exine exhibits a fascinating array of Patterns and design. Why do you think exine should be hard? What is the function of a germ pore? The inner wall of pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of a pollen grain is surrounded by a plasma membrane. The pollen grain is mature. It contains two cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve and a large, irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, the pollen grain are shed at this two-cell stage. In the remaining species, the generative cell mitotically gives rise to two male gametes before pollen grains are shed. The three-cell stage. Pollen grains are of many species, cause several layers. Let's end this first. They give the structure of the pollen grain. Generally spherical, diameter of 25 to 50 micrometer, two layers of intine, exine and intine. Exine is made up of sporopollenin, of the hardest or the most strongest materials or hardest materials known, which is yet no enzyme that degrades sporopollen has been found, resists high temperature, high pH, extremely high or extremely low pH, acidic or highly alkali or highly acidic mediums. Meaning, what they meant to say is sporopollenin is really resistant, and this sporopollenin, on the pollen grain there are pores which we call as germ pores, which is where the pollen tube arises from, and this is the exines. Due to exine itself, pollen grains can be preserved for many years due to the presence of sporopollen. The exine layer's main function: protection of the pollen grain, protect pollen grain. Second function, germ pores which help in germination of the germinate pollen tube. These are the two main functions of the exine layer, which is made up of a really resistant sporopollenin. And if we go with intine, then intine, in exine we say that it was non-continuous and germ pores were there, but intine is the inner layer which is continuous or smooth. It is made of cellulose and pectin, and it completely covers the whole pollen cell. This is the intine. Also acts as a protection. That is with the pollen grain. Yes, and as for pollen division, this is the microspore tetrad. Here, this microspore tetrad, as the anther matures and dehydrates, this is a single pollen cell right after the microspore tetrad is divided. This is the cell. This is after some time where two vacuoles and an asymmetrical spindle appear, and This nucleus will divide into two cells of the vegetative cell and the generative cell. The vegetative cell has a large irregularly shaped nucleus and it has nutrient rich cytoplasm, and whereas the generative cell is small, the nucleus of the generative cell is spindle shaped, and this cell will float in the vegetative cell, float in the cytoplasm of the vegetative cell. After some time, after this completes 60% of the angiosperm. This is what we call the, when there is one generative cell and one nucleus, this is what we call the two cell stage. And in 60% of the angiosperms, the anther will dehiscence and release the pollen grain at this stage itself. But in 40% of the other species, in the anther itself, the generative cell, this cell, will mitotically divide to give two gametes. These are the male gametes, and this is what we call the three-cell stage. And that is the structure of the pollen grain, and the uses and applications or harms caused by pollen grains. Pollen grains of many species cause several allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. Pollen grains cause, as we saw, there are different types of pollen grains, and we saw there were spiky ones, ribbon like ones, and etc. These pollen grains will cause, if inhaled, will cause severe allergies of bronchial afflictions. Bronchial meaning the bronchi as the nasal cavity, trachea, and bronchi to primary bronchioles, secondary, tertiary bronchioles. This is the respiratory system. In this, it will call bronchial afflictions, maybe irritations, and in some people often leading to chronic respiratory disorders like asthma and bronchitis. Chronic meaning long term, acute meaning short term disorders. Cold is an acute disease which will end in a week or so, but chronic meaning it will last for a long term or even a lifetime. This is what we learned in lower classes. It may be mentioned that Parthenium or carrot grass is another name of Parthenium that came into India as a contaminant with imported wheat, has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. Has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, as we saw before, the pollen cell is divided into the vegetative and the generative cell, and we saw that the vegetative cell's nucleus is nutrient rich. As such, it has become a trend in Western countries to consume this pollen as pollen tablets. A large number of pollen products in form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. Remember that it has only been claimed. This is the pollen products. Bee pollen from England. This is the tablet. Pollen products such as tablets, syrups is believed to be nutritious and increase the performance of athletes and racehorses. Well, let's continue reading. When once they are shed, pollen grains have to land on the stigma. Another concept we are bringing here, it is viability. Viability of pollen grains. After the pollen grain, after the dehiscence of the anther, this is an anther, this is the pollen grains, pollen out. After this, how much time after should this pollen, how much long will this pollen grain, if pollen grain should land on stigma, then only they can germinate a pollen tube and fertilize the ovules. For how much time this pollen grain can remain like this? If after does this pollen grain for like after 30 minutes, if we keep it on the stigma, will it still germinate the pollen tube? What about 2 years? What about 3 years? If this pollen grain it will land on the stigma after release, how many years or how much time will it last? How much time will it remain viable? Viable meaning if we keep that pollen on the stigma, it should germinate into a pollen tube. After till how much time will this pollen will be fresh that if we keep it on a stigma, it can generate the pollen tube. That is the concept of viability of pollen grains. This will differ in different species of plants. Let's continue reading. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. And in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. You may have heard of storing semen/sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of our species for years in liquid nitrogen -196 degrees. Such stored pollen can be used as pollen banks similar to seed banks in crop breeding programs. So basically what they are saying is this viability in some cereals, like rice, wheat, it is found that the pollen will lose viability within 30 minutes, such a low time. In 30 minutes after the pollen is shed, in 30 minutes the pollen doesn't land on stigma, then that pollen, even if it lands on stigma after 30 minutes later, it won't germinate into the pollen tube. In the second type, in some members of Rosaceae, Leguminosae, pea family, and Solanaceae, potato family, the viability will remain for months, even months after this kind of family members. Some members, even after several months, if you take the pollen after it is shed and keep it on a stigma, there will be germination and fertilization. These are some examples they give, and we can tell that cereals and rice, wheat generally have low viability period, whereas Rosaceae, Solanaceae and Leguminosae members have long viability period. And The next being these pollen grains, to avoid loss of viability, after some time they will lose viability, but to avoid that loss of viability, can be stored in containers of liquid nitrogen having temperatures of minus 196 degrees Celsius of liquid nitrogen. In these containers, if these pollen grains are stored, they can be stored without losing viability for years. This process is what we call cryopreservation. These pollen grains can be used as pollen banks, such as seed banks, when used during fertilization for used for purposes of fertilization. This is in crop breeding programs. Now the second part, which is the gynoecium, the pistil, megasporangium, ovule, and embryo sac, which is the gamete or megaspore. Megasporangium is ovule and megaspore is embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary. Let's read again. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. There are more than one pistils. The pistils may be fused together, syncarpous, or may be free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is elongated slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, the locule. The placenta is located inside the ovarian cavity. So they describe the structure. There are many types of pistil, carpel, and etc. The first type, under pistil, there are two types. One is monocarpellary. Monocarpellary meaning having single flower having only one pistil or one carpel. Second is under pistil. The division is monocarpellary or multicarpellary. Multicarpellary meaning a flower having more than one pistil. And in this multicarpellary, there are again two divisions of the syncarpous and the apocarpous. Apocarpous is when ovaries or the carpels are free, not fused, and in syncarpous it is fused carpels. This is the division. There are two types of flowers, which are mono or multicarpellary flowers. Monocarpellary flowers have a single carpel, whereas multicarpellary flowers have more than one carpel. Multicarpellary carpels are divided into two types of syncarpous and apocarpous, where syncarpous is the fused carpel and apocarpous is the free carpels. Example for syncarpous and apocarpous. For apocarpous, it is Michaelia champaka. They have given the diagram here. This is the champaka Michaelia flowers, multicarpellary apocarpous condition. A multicarpellary apocarpous gynoecium of Michaelia champaka. And for for a syncarpus flower, syncarpus pistil of showing the syncarpus pistil of papaver. So example based on NCERT, we can take syncarpus as papaver. Papaver is an example for syncarpus ovary, whereas for apocarpus or free carpels, this is the syncarpus fused ovary, which is the papaver plant, and this is the Michaelia plant, where individual ovaries, not ovaries, individual carpels are separated like this. This is the apocarpus condition, where we can take the champaka flowers or the Michaelia champaka. Michaelia champaka. And the gynoecium is divided into, this is the stigma, which is a landing platform for microspore or pollen grains. This is called as a stigma. This long slender stalk is what we call style. This bulge portion at the end is what we call ovary, and the base of the flower is what we call thalamus. The ovary contains an embryo sac. This is the ovule, and ovary contains ovules. They can be arranged in various arrangements, which we call placentation. The arrangement of ovules inside the ovary, inside the locule of the ovary, the space inside the ovary is called locule, and the arrangement of ovules inside the ovary is what we call placentation. We saw that there were many types of placentation of the axial, parietal, marginal, etc. in the first PU chapter. Placentation is the arrangement of ovules inside the ovary. And this is the diagrammatic view or section view of a an anatropous ovule. Anatropous being this funicle is in the same plane as the micropyle. Micropyle and funicle are in the same plane, which leads to us calling it a anatropous ovule. This is the ovule. The ovule has a stalk which connects it to the ovarian wall, which is called funicle. And the connection between the ovule and the funicle leads a mark on the seed, which is called hilum. The opening of the ovule where the pollen tube should enter is called micropyle. This region where micropyle is present is called the micropylar region or the micropylar pole, and the opposite to that is what we call the chalazal pole or chalazal region. The ovule is covered by two integuments of the inner and the outer integuments, where they will later develop into testa and tegmen in the seed. As ovule will develop into the seed, this inner and outer integuments will develop into testa and tegmen, which are the seed coats, seed layers of the seed coverings. And this is the embryo sac. Nucellus is a nourishment tissue which will nourish the embryo. This nucellus is in similar function to the tapetum cells, which we saw in the androecium, similar to tapetum. Now let's continue reading. I dissected flower of hibiscus. Yes. Class 11, arising from the placenta are the megasporangia, commonly called as ovules. The number of ovules in an ovary may be 1, wheat, paddy, mango, to papaya, water, to many in papaya, watermelon, and orchids, with orchids having the highest number of ovules. The megasporangium or ovule, let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovules is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represent the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus, except at the tip where there is a small opening called micropyle is organized. Opposite to the micropylar end is a chalazal end, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called a nucellus. Cells of nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gamete. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis, the process of formation of megaspores from megaspore mother cell. The topic for this is basically explaining the structure of the anatropous ovule. And the next topic is the megasporogenesis. Let's read the topic again. Megasporogenesis, a process of formation of megaspore from the megaspore mother cell MMC is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell MMC in the micropylar region. This is the different stages. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cells in the micropylar region of the nucellus. It has a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of MMC undergoing meiosis? Meiosis results in the production of 4 megaspore, female gametophyte in majority of the flowering plants. One of the megaspores is functional while the other 3 generate. Only the functional megaspore develops into a female gametophyte. Basically, let's assume this is a megaspore mother cell. A megaspore mother cell. This is 2N ploidy. This cell will undergo meiosis near the micropylar region. This megaspore mother cell will undergo meiosis in the microspore region to obtain four identical gametes. These are all N. These are the four cells which are obtained from the megaspore mother cell get undergoing meiosis. This of these four cells, three will degenerate. Three cells, for example, let's take these three. These three will degenerate and only one cell will be remained. This is the nucleus of the cell that remains. This is what we call where out of the four cell produced, only one cell undergoing, one cell remaining. This is called monosporic development. This is not seen in every species of plant, but majority of the species. This is what we call monosporic development, when out of the four cells produced, only one will continue to develop, whereas the other three will degenerate. This is the first stage. After this is a cell, this is a single cell which is as N ploidy, and in this cell the nucleus will continue to undergo division. The nucleus will undergo division mitotically, producing two nucleate stage, four nucleate stage. This is mitosis of nucleus. Four celled, and finally it will stop at the 8 cell stage, where three cells will be here, three nucleus will be here at the chalazal end. These cells is what we call antipodal cells, and at the center there will be two polar nuclei, is called the polar nuclei, central polar nuclei, and here there will be another three nucleus will be there, of which two will form the synergids and one will form the egg cell. This is the egg cell. These both are the synergids. If we take the enhanced view of a synergids, there will be, these are the synergids, let's assume. These are the synergids. This is an enhanced view. This is the nucleus. This is the egg cell. Here there will be a finger-like projection or a special projection here. This is the micropylar end. Synergids will be present at the micropyle end. Antipodals will present at the chalazal end. And this filiform apparatus, this apparatus, finger-like projections, is what we call the filiform apparatus. The filiform apparatus. This filiform apparatus helps in guiding this is the micropolar region. Pollen tube will enter through here. This filiform apparatus will guide this pollen tube inside the ovule. This is the microsporogenesis, and this is the final, what we call the mature embryo sac. mature embryo sac, I'll draw the mature embryo sac again. Basically, let's see the megasporogenesis once again. Megasporogenesis, where the megaspore mother cell having a ploidy of 2n will undergo meiosis and produce 4 cells, 4 haploid gametes, of which 3 will degenerate, 3 degenerate, and 1 will continue to develop. This is called monosporic development. And in this one cell, the nucleus will undergo mitosis to have the 2 nucleate, 4 nucleate, and finally the 8 nucleate and 7 cell stage. I will draw this stage is what we call a mature embryo sac. Yes, now I will draw the mature embryo sac. Mature embryo sac. This is the chalazal end. This is the micropylar end. Of which these cells are antipodal cells. The center we have 2 nuclei which we call polar nuclei, and here this is the egg cell, synergids. This is the filiform apparatus which will help in guiding the pollen tube. The 3 cells of the egg cell, the synergids, 2 synergids, 1 egg cell and 2 synergids, the total we call it as egg apparatus. This is what we call as egg apparatus. This is the mature embryo sac. Now let's go back. Now that is the basics of the microsporogenesis. The same diagrams are drawn here. This is a megaspore mother cell. This is a new cell as they highlighted. This is the micropolar end, megaspore mother cell. Megaspore first will undergo meiosis. This is the after undergoing meiosis 1, it will have 2 cells, this one cell and this one cell, and after undergoing this meiosis 1 and after undergoing meiosis 2, you will have 4 cells, of which 3 degenerate and only one cell will remain. That cell will be like this, and from this stage the nucleus will start mitotic division. This is the 2-cell stage, 4-cell stage, 4 nucleate stage, 2 nucleate stage and 4 nucleate stage. The nucleus will undergo continuous mitotic division, where this is the 2 nucleate stage, 4 nucleate, this is the final 8 nucleate stage. This is the whole formation of this is the full mature embryo sac. Where these are the antipodals, polar nuclei, central cell, egg, synergids, filiform apparatus. This is what we call the mature, a diagrammatic representation of the mature embryo sac. And let's continue reading. Of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. This they are talking about MMC, megaspore mother cell. It has a nucellus, a large cell containing dense cytoplasm and a prominent nucleus. MMC undergoes meiotic division. What is the importance of MMC undergoing meiosis? Meaning, the MMC is a diploid cell, but gametes should be haploid, right? That is why it will undergo meiosis. Meiosis results in the production of 4 megaspores, female gametophyte. In the majority of flowering plants, one of the megaspores is functional while the other 3 degenerate. Only the functional megaspore develops into female gametophyte, embryo sac. This method of embryo formation from a single megaspore is termed as monosporic development. What will be the haploidy of the cells of nucellus, MMC, the functional megaspore and the female gametophyte? The cells of nucellus will be 2n, MMC will also be 2n, megaspore, functional megaspore will be n, as it is after meiotic division. The female gametophyte, which is the embryo sac, will also have a ploidy of embryo sac ploidy will be having 8 nucleate, but as the main fertilization is going to happen, embryo sac will be 4n, but the egg cell where the the syngamy happens will have only n. Female gametophyte is embryo sac. As such, it will have 4n ploidy. Now they are giving a discussion of what I just said. Let us study about the formation of embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form 2 nuclei, which move to the opposite poles, forming 2 nucleate embryo sac. 2 more sequential mitotic divisions result in the formation of 4 nucleate and later 8 nucleate stages of the embryo sac. It is of interest that these mitotic divisions are strictly free nuclear, that is, only nuclear divisions are followed. Only nucleus will divide. They are only free nuclear, meaning only nucleus will divide. Nucleus divide. There is no division in cytoplasm or the cell. Nucleus divide. cytoplasm or cell don't divide. Yes. Immediately by cell wall formation. After 8 nucleate stage, cell walls are laid down, leading to the organization of typical female gametophyte of the the embryo sac. Observe the distribution of cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in a large central cell. Here we see that antipodals, each nuclei will form 3 antipodals, so 3 nuclei will form 3 cells of the antipodal cells, and here 2 synergids will take 2 nuclei and 1 egg cell will take 1 nuclei. But in the center there are 2 nucleus. This nucleus will together form a large central cell. So the ploidy of the central cell will be 2n, whereas the ploidy of antipodals and the synergids along with the egg cell will be n. So yes. Let's continue. There is a characteristic distribution of these cells within the embryo sac. 3 cells are grouped together at the micropolar end and constitute the egg apparatus, the 2 synergids and 1 egg cell. The egg apparatus in turn consists of 2 synergids and 1 egg cell. These synergids have special cellular thickenings at the micropolar tip called filiform apparatus. This filiform apparatus are special cellular thickenings. That is what filiform apparatus is. Now, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazal end and are called antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei, thus a typical angiosperm sac. At maturity, though 8 nucleate is called 7 celled, which is called the 8 nucleate and 7 celled stage. Now let's come to the next main topic, pollination. Pollination is divided. The topic is pollination. Pollination is divided into 3 types. 1. Autogamy. 2. Geitonogamy. 3. Xenogamy. Autogamy is pollination of a flower from its own pollen grains. Pollination of a flower from its own pollen grains is what we call autogamy. Geitonogamy is pollination of a flower from another flower in the same plant. So the conditions here are autogamy being pollination of a flower from its own pollen grains, meaning autogamy, the flower must be bisexual, meaning it should have both the androecium and the gynoecium. Autogamy is only possible when the flower is bisexual. The mandatory condition for autogamy is flower should be bisexual, as the pollination of a flower happens from its own pollen grains. This is the flower, this is the stigma, or the stigma is pollen grains. These pollen grains will come and land on the stigma and pollinate the flower. This is the autogamy. Whereas in case of geitonogamy, the flower may not be bisexual. There is no mandatory condition that flower might or might not be. Flower need not be bisexual in geitonogamy. In geitonogamy, flower need not be bisexual, but the plant should for sure be bisexual, or in another term, monoecious, or in another word, hermaphrodite. The flower need not be bisexual, but the plant which bears the flower should bear both male and female flowers. In only such conditions, geitonogamy is possible. Geitonogamy is when, if this is a plant, these are flowers, and this is a male flower, now if these are the two flowers, this is a single plant, it has two flowers, and this is a male flower, this is a female flower, this is the anther, I mean stamen, this is the stigma. This pollen grain will come and fertilize this plant, this flower. This is the case for geitonogamy, not xenogamy. I'll explain xenogamy in the next page. So basically, autogamy condition is flower must be bisexual, and it is pollination of a flower from its own pollen grains. This is autogamy. And geitonogamy, the flower need not be bisexual, but the plant should be bisexual. It should be monoecious, bisexual, or hermaphrodite. So basically, we see that in autogamy and geitonogamy, the common thing is the same plant, same flower. The flowers in the same plant are pollinating each other. So genetically, the genes from the same plant are used for fertilization. So autogamy Autogamy and autogamy and geitonogamy are genetically similar, but physically different. The genes which we get in both autogamy and geitonogamy, the flower pollinated will be from its own flower, or the flower from its own plant. So it is genetically similar but physically different. But as physically it is comes under cross pollination. Geitonogamy under physically comes under cross pollination, but genetically is same as autogamy or self pollination. This is autogamy and geitonogamy. Let's go to xenogamy. The third type of fertilization is xenogamy. Xenogamy is when two flowers from two different plants of the same species fertilize each other. Two flowers from two different plants of the same species fertilize each other. This is xenogamy. Xenogamy. Two flowers from two different plants of the same species fertilize each other. Xenogamy, there are different types of xenogamy, and under xenogamy, which is called cross pollination, both genetically and physically, yes, this is the pollination ways. This is done with pollination. Yes. Now let's continue reading. In the preceding section, you have learned that male and female gametes in flowering plants are produced in the pollen grain and the embryo sac, respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Let's read again. Pollination is the mechanism to achieve this object. Pollination is a mechanism. Okay. Transfer of pollen grains. Transfer of pollen grains shed from anther to stigma of pistil is what we call pollination. Kinds of pollination. Depending on the source of pollen, the classification on kinds of pollination is dependent on the source of pollination. Based on the source of pollination, first division, autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from anther to stigma of the same flower. In a normal flower, pollination is achieved with the flower. Transfer of pollen grains from the anther to stigma of the same flower. Pollination is achieved within the same flower. Transfer of pollen grains from anther to stigma of the same flower. That is autogamy. In a normal flower which opens and exposes anthers and stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity as well. The anthers and stigma should be close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis and commelina produced two types of flowers: chasmogamous flowers, which are similar to flowers of other species with exposed anthers and stigma, along with cleistogamous flowers which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anthers dehiscence, the flower bud's pollen grain come into contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous, as there is no chance for cross-pollination. Cleistogamous flowers produce assured seed set even in the absence of pollinators. Do you think the cleistogamy advantages or disadvantages to the plant? Why? So, autogamy, for these conditions are: flower must be bisexual. Second condition, anther and stigma should lie close to each other. Third condition, the timing of dehiscence of anther and receptivity of stigma should be same. Only in such conditions where we see autogamy occurs. In example, in some flowers like viola, common name is common pansy, commelina, in these flowers, these plants produce two kinds of flowers. One, chasmogamous flowers, which are the normal flowers, and for chasmogamous flowers we need these conditions. These conditions apply to the chasmogamous flowers. And two, cleistogamous flowers. These cleistogamous flowers are special bisexual flowers where the petals or the flower won't bloom. In these flowers, there's 100% chance of self-pollination or autogamy occurring, as there is no chance for cross-pollination unless done artificially. %100% chance of autogamy and 0% chance for cross-pollination, that is in cleistogamous flowers, as the flower doesn't bloom itself. It will be completely closed. And as for advantages of the autogamy, is that advantages, if we think of advantages, one, there will be no genetic variation in the seeds produced, so we can call it as a assured seed set, as how the plant will be there exactly similar, the seed, the plant produced by the seed will also be there in case of cleistogamous flowers, because there is no genetic variation at all. Second, no need for pollinating agents. We don't need anyone's help for the pollination here. It will automatically happen as long as the anthers, dehiscence and stigma receives the pollen grain. No need for pollinating agents. No need for separate sector for helping in pollination. These are the advantages. As for disadvantages, one, no genetic variation. It may be a disadvantage, may be an advantage as well. No genetic variation, there it will lead to no genetic variation will lead to less adaptation. Sexual pollination, sexual reproduction is mainly occurs because for genetic variation itself, for more adaptation, sexual reproduction occurs. If here what is happening is what is similar to asexual reproduction. So using sexual reproduction, reproducing organs for asexual reproduction, genetically, what is the use? There is no use, no genetic variation, less adaptation. This is the main disadvantage. This is the autogamy. Now, two types of flowers, Lysto and chasmogamous flowers. These are the flowers. This is seen in common area, I believe. This is, they didn't give which plant. Yes. These are the chasmogamous flower. There is a blooming here. The flower bloomed here. Whereas here, this is the cleistogamous flower. Whereas there is no blooming only. They did not bloom itself. These are the cleistogamous. These are the chasmogamous flowers. So Geitonogamy. Geitonogamy, transfer of pollen grains from the anther to the stigma of another flower of the same plant. Same plant, different flowers. Although geitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollination comes from the same plant. Xenogamy, transfer of pollen grains from anther to stigma of different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. And it is xenogamy is what is actually mostly preferred in the plants. Agents of pollination. Plants use two abiotic, wind and water, and one biotic, animal agents to achieve pollination. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic agent. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainties and associate loss of pollen grains, the flower produce enormous amount of pollen grains when compared to normal ovules, number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also requires the pollen grains are lightweight, non-sticky, so that they can be transported in wind currents. They often possess well exposed stamens so that pollen are easily dispersed into the wind currents, and large, often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers are often have single ovule in each ovary, and numerous flowers are packed in the inflorescence. A familiar example is the corn cob. The ears you see are nothing. The ears you see are nothing here. This part is nothing but stigma and style which wave in the wind to collect or trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall the water is a regular mode of transport for male gametes among the lower plant groups such as algae, bryophytes, and pteridophytes. It is believed that particularly for some bryophytes and pteridophytes that their distribution is limited because the need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria, Hydrilla, Zostera as well, grow in fresh water, and several marine seagrasses such as Zostera. Not such as water hyacinth and water lily. Water hyacinth and water lily are not water pollinated plants. The flowers emerge above the level of water and pollinated by insects or wind, as in most of land plants. Vallisneria, the female flower, reaches the surface of water by long stalk, and male flower or pollen grains are released to the surface of water. They are carried passively by water currents. Some of them eventually reach the female flowers and the stigma in other group of water pollinated plants such as seagrasses. Female flowers remain submerged in water and pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most water pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. So now basically xenogamy. The first type is pollination by wind, which we call it as anemophily. Anemophily, wind pollination. Before that, just a brief view of what is pollination. Pollinating agents. Pollinating agents are primarily divided into 1. Biotic, 2. Abiotic. Biotic is by animals. Abiotic is by wind or water. But basically, most of the angiosperms prefer biotic pollinating agents, which are animals, insects, etc. Most preferred. abiotic is less preferred, and in abiotic, wind pollination is more preferred. And water pollination is mostly less preferred in angiosperms, with mostly limited to 30 genera, as water pollination is more seen in less plants of the pteridophytes, bryophytes, etc. In the lower plants we see more water pollination, and it is believed that because of the dependence on water pollination, most of the bryophytes, which depend on mainly water pollination, are limited because water pollination success rate is really low, as it is mostly a chance factor. Basically, there are two kinds of pollinating agents, biotic and abiotic. Biotic agents is mostly preferred by plants, and abiotic is divided into wind and water. Wind is more preferred compared to water. Wind is what we call anemophily or wind pollination. Let's start. Anemophily or wind pollination. Wind pollination, it is the flowers seen here. Flowers opting for wind pollination, we see that one, they lack nectar, fragrance, and attractive colors, and are inconspicuous, as they don't need to attract any insects or any other animals towards them. They do not provide nectar, the pollen gift, pollinate for the pollinators, fragrance, attractive colors, and are mostly inconspicuous, which means they cannot be identified very easily. As such, like normal flowers, we can easily from far as well we can identify those are flowers and those are beautiful. But these flowers pollinated by wind, are inconspicuous. They lack nectar, fragrance, and attractive colors. 2. The pollen grains here are non-sticky, lightweight, and are easily transportable in wind currents. This is the second adaptation. The pollen grains, being non-sticky and lightweight, are easily transported in the wind. And the third adaptation is that more number of pollen grains produced. Enormous amount of pollen grains will be produced compared to ovules. Compared to ovules, the ratio between the number of ovules and number of pollen grains will be inside of the pollen grains, as enormous amount of pollen grains will be produced. As this wind pollination depends on a chance factor, the number of pollen grains should be increased to compensate the less chance, as if more pollen grains are produced, more chance of pollination. As such, the ratio between pollen grains or microspores and megaspores, it will completely bend into the side of the microspores or pollen grains. and of course the flowers containing less number of ovules, which most flowers containing only one ovule. Pollen grains will be large amounts, flower will contain only one ovule. This is the third point, third adaptation. The fourth adaptation, if we see, the fourth adaptation is yes, the adaptation of stigma and styles. If we see in corn cob, this is the corn, we see number of feathery hair-like structures. These hairs are what we call tassels. These tassels are nothing but stigma and style. They are in this feathery adaptation because they can easily trap the pollen grain which are coming and get fertilized. This adaptation is to trap the pollen grains, to catch the pollen grains. These are the four main adaptations seen in the flowers pollinated by wind. 1. They lack nectar, fragrance and attractive colors and are inconspicuous. The pollen grain produced are non-sticky, lightweight and easily transported in the wind currents. They produce more number of pollen grains compared to ovules and less number of ovules. Only one ovule in most of the flowers. This is a adaptation seen in corn cob, which we see tassels, which are feathery stigma and style, which help in catching of the or trapping of the pollen grains, which are transported via the wind. These are the main adaptations seen in wind pollinated flowers. Next is hydrophily or water pollination. In water pollination, these water pollinated flowers, we can take an example of Vallisneria, Zostera, which are seagrass, and one more being Hydrilla. These are the actual water pollinated plants, and this in abiotic category, the water pollination plants are limited to less than 30 genera, meaning it is less opted for. In Vallisneria, we see a special pollination mechanism where if this is the water surface, the anther, the stigma will be near the surface of the water, and the male flowers will come near, or the pollen grains will come near and fertilize the stigma or flower. Normally the Vallisneria will be highly coiled and submerged in the water, the female organ, and later it will be released, the coil is released and it will reach the surface of the water, and male flowers will approach this female organ, female gynoecium, and the pollen will fertilize the stigma. This is seen in Vallisneria. In Zostera and seagrasses, monocotyledons, It is also seen mostly in monocots. Wind pollination is also seen in grasses, monocots. Wind pollination as well as water pollination is more seen in monocots and grasses. This is also for wind pollination, anemophily. And normally the adaptation, this is one adaptation we see in Vallisneria, and one more adaptation is the pollen grains in submerged plants will be ribbon like to allow easy passive transport in water the pollen grains will also be covered by a gelatinous sheath or mucilaginous sheath. This will prevent the pollen grains from getting wetting or degrading from the water, continuous water exposure. Gelatinous sheath, as they will be continuous to exposed to the water, this mucilaginous sheath will prevent the degrading of the pollen grain, and they will be ribbon shaped to easily be transported in the water currents. This is the two adaptation we see in water pollinated submerged plants, and a difference from the why water hyacinth and water lily, lotus are not even though they are aquatic plants, not water pollinated plant, not opt for or not water pollinated plants is because they are not water pollinated plants, because generally if we see a lotus, it will be floating. Don't mind the drawing. Let's assume this is a lotus. This will be floating on the water. Let's assume this is the water surface. This is a lotus. It will be floating on the water, and only the roots will be submerged in the water. As such, even if there is a pollen grain, it cannot reach the stigma like this. So lotus is often insect pollinated plant, insect pollinated or wind pollinated. And not water pollinated. Now let's continue reading. They asked that in most of the water pollinated species, pollen grains are protected from wettiness. Both wind and water pollinated plants are not very colorful and do not produce nectar. What would be the reason for this? Because they mainly opt for wind and water pollination, and they don't need to attract insects. Insects are attracted by nectar and flowers, but since they are wind pollinated and water pollinated, they don't need these insects. As such, they don't need to produce nectar, which is the pollen reward or reward for the pollinators, as wind and water are abiotic. They don't need pollen or nectar. Now let's continue reading. This is the mechanism. This is the Vallisneria flower, where it will reach the surface. The female gynoecium stigma will reach the surface. This is the female flower, and it will reach the surface of the water, and male flower will come near here. This is the male flower. So this is minute, but this is the male flower. This is the enlarged view of the male flower. It will come near and fertilize the or pollinate the female flower. The female flower, as you see here, is highly coiled and will be like this. During time of pollination, it will uncoil and reach the surface here. This is seen in Vallisneria. And as for more diagrams here, this we will see this. This is the pollination by insects, and in the insect we see that the highest percentage we see it is biotic pollination, most preferred in plants. Biotic pollination, most preferred. In biotic pollination, entomophily, insect pollination, most preferred. Biotic pollination is preferred over abiotic pollination. In biotic pollination by animals, insect pollination is more preferred. In insect pollination, bee pollination percentage is most. In biotic pollination is more preferred. In biotic pollination, insect pollination is more preferred. And in insect pollination, bee pollination is the most preferred by flowers. Now let's continue reading. Majority of flowering plants use a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds, and bats are common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptiles, gecko, lizard, and garden lizard have also been reported to act as pollinators. So they're saying that many of the flowering plants, mostly we see that it is pollinated by bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, hummingbirds, and bats are common pollinating agents. Among these, insects, particularly bees, are the dominant biotic pollinating agent. Even larger animals such as some primates like lemur, arboreal, tree-dwelling rodents, or even, and in primates, lemurs, rodents, or even reptiles like gecko, lizard, and garden lizard have also been found to act as pollinators in some species. Lemur is an example for a primate pollinator. Often, flowers of animal pollinated plants are specifically adapted for particular species of animal. In pollinated by biotic agents, they are adopted for only some species of animals, not all. They are specialized or a particular species of animal. Majority of insect pollinated plant flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. In wind pollinated plants, we saw that the flowers were inconspicuous, but here, to attract insects, if the flowers are small, they are formed into clusters to make them conspicuous. Animals are attracted to flowers by color and fragrance. The flowers pollinated by flies and beetles secrete foul odors to attract these animals or insects. To sustain animal visits, flower has to provide rewards. These are called floral rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitor comes in contact with anthers and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carries the pollen on its body, comes in contact with the stigma, it brings about pollination. In some special flower rewards are in providing safe places to lay eggs, as example that of the tallest flower of Amorpha phallus. The flower itself is about 6 feet in height. A similar relation exists between the species of moth and the plant yucca, where both species, moth and yucca, cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary, and the flower, in turn, gets pollinated by the moth. The larvae of the moth come out of eggs as the seeds start developing. So, basically what they're saying is, flowers pollinated by animals are specialized for a certain species of animals. For example, in insect pollinated plants, flowers are large flowers, colorful, fragrant, conspicuous, and if small flowers are there, they will be clustered to make it look beautiful. And in the pollen grain adaptation, we saw that in wind pollination, the adaptation was that the pollen grains were non-sticky and lightweight, but here the pollen grains are sticky. And for example, another type where it is in flowers pollinated by flies or beetles, these flowers secrete foul odors etc. These are the adaptations seen based on the specific requirement of the flower. Now, the next topic is about the floral rewards. As the flower needs the pollinators to pollinate, to help in pollination, they provide floral rewards. As if the insects stop getting attracted by the flowers and do not help in pollination, the plant species might get extinct, which solely depend on insect pollination. As such, they need to attract these insects to help in pollination, and as such they provide rewards which are called floral rewards. FLORAL rewards. These can be 1. Nectar, 2. Pollen grains, and in some special cases safe places for insects to lay eggs. Example, in the Amorphophilus plant, the flower is 6 feet tall. As it is of such high height, it is relatively safe. And in another life cycle, we see that there's another example they gave in textbook, which is in case of yucca plant and I think they gave the yucca plant pollinated by moths. Yes, and the moths. In this case, these both are interdependent on each other, where both life cycles cannot be completed if one is absent. Basically, yucca plant can be pollinated by moths. It will be a closed flower. It will be a flower, stigma, and the moths taking pollen will lay their eggs inside the yucca flower and also pollinate the flower. This is a mutual relationship where moths will get a safe place to lay eggs, and yucca plant, providing the safe place, will get pollinated by the moth. A mutual relationship between yucca plant and the moths. Let's continue. Why don't you observe some flowers of the flowering plants or any others available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobelia, cotton, tobacco, rose, lemon, and eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You'll have to patiently observe the flowers over a few days and at different times of day. You could also try to see whether there is any correlation in the characteristics of a flower to that of to the animal that visits it. Carefully observe if any of the visitors come in contact with the anthers and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as nectar robbers or pollen robbers. You may note that you may not be able to identify the pollinators, but you will surely enjoy your efforts. Basically, the pollinators, the insects or other visitors which eat the pollen or take the nectar but do not help in pollination are what we call pollen or nectar robbers. Next is the outbreeding devices. Outbreeding devices. Majority of the flowering plants produce hermaphrodite, meaning monoecious or bisexual flowers, and pollen grains. Hermaphrodite, write the meaning. Hermaphrodite meaning bisexual. And pollen grains are likely to come in contact with stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive or stigma becomes receptive. Or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that pollen cannot come in contact with the stigma of the same flower. Both these developed devices prevent autogamy. The third device is to prevent inbreeding is self-compatibility. This is a genetic mechanism and prevents self-pollination pollen from same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device is to prevent self-pollination is to production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, such as monoecious or bisexual plants, it prevents autogamy but not geitonogamy. In several species, such as papaya, male and female flowers are present on different plants. This is each plant is either male or female, dioecious. This condition prevents both autogamy and geitonogamy. Let's read again. Majority of flowering plants produce bisexual flowers and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive or stigma becomes receptive much before the release of pollen grain. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding depression is self-incompatibility. This is a genetic mechanism and prevents self-pollen from same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination of pollen growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, it prevents autogamy but not geitonogamy. In several species such as papaya, male and female flowers are present on different plants, that is, each plant is either male or female. The condition prevents both autogamy and geitonogamy. All plants, or not all but many plants, prefer sexual reproduction xenogamy. This is because they need genetic variation. But because this is genetic variation and xenogamy is preferred by many plants, but if but many of the angiosperm plants produce bisexual flowers. Both androecium and gynoecium will be there in the same flower. In such cases, autogamy chances are there. Even though the plant dislikes autogamy, because it's producing bisexual flowers, there are high chances for autogamy itself. And if this process of autogamy repeats many times, the plant will have an inbreeding depression. Inbreeding depression. To prevent this, devices to prevent autogamy in bisexual flowers or in these plants, many adaptations were made in the plants. One such adaptation is making the time of dehiscence of anther and time of receptivity of stigma will be different. If such a case, even if the pollen is released and it will land on the stigma, that by the time the stigma gains receptivity, the pollen will already be will already have lost its viability. As such, this is one of the adaptations seen to prevent inbreeding depression. And the second one is the androecium and gynoecium will be separated by physical space or some separations. This is the second adaptation. Third is a genetic mechanism, which is self-incompatibility. Where both geitonogamy and autogamy is prevented genetically. If the pollen from the same plant falls on the same flower, there is no, if same pollen pollen from same plant or same flower falls on stigma, the stigma will not allow pollen tube generation. Will not allow, falls on stigma, the stigma will not allow pollen tube generation. This is self-incompatibility. And the next one, let's read this again. Many plants prefer xenogamy over auto or gaetanogamies. This is due to genetic variation, but many plants produce bisexual flowers. In such cases, autogamy chances are there. To prevent and if there is a repeated autogamy, there is a chance for cause of inbreeding depression. To avoid this, the adaptations in plant, 3 adaptations are listed here. This is the time difference between the dehiscence and the receptivity of the anther and stigma respectively. It will be different. This condition is called protoandry, or when andricium will the anther will dehiscence before, or protogyny. I don't remember the exact name, but it should be something like this. When the stigma will gain receptivity before the dehiscence of anther. And the second adaptation is the andricium and gynoecium will be separated by physical space or some separations. The third is genetic mechanism. Self-incompatibility. If same pollen falls on stigma, so if the pollen from same flower or same plant falls on the stigma, then the stigma will not allow the pollen tube generation. This is called self-incompatibility. The fourth adaptation we see is production of unisexual flowers. In case of castor maize, we see unisexual flowers, but the plants themselves produce both male and female flowers. The plants are bisexual. The flowers are unisexual, but plants are bisexual. In such case, we can prevent autogamy. There is a 100% chance to prevent autogamy, but not gaetanogamy. Because the different flower, male and female flowers of the same plant will fertilize. The male flowers of the plant will fertilize the female flowers in the same plant. But complete prevention is seen in example of papaya. In papaya, the plants themselves produce only male or female flowers. The plants are dioecious or unisexual. The plants can only produce either male or female flowers. In such cases, both autogamy and gaetanogamy are prevented. Let's continue. The next topic is pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also lands on the stigma. The pistil has the ability to recognize the pollen. The pistil has the ability to recognize the pollen, whether it is the right type, compatible, or the wrong type, incompatible. If it is the right type, the pistil accepts the pollen and promotes pollen pistil events. Promotes post pollination events that leads to fertilization. That is when the pollen is compatible. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in style. The ability of pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and pistil. This dialogue is mediated by chemical components of pollen interacting with those of the pistil. It only in recent years that botanists have been able to identify some of the pollen and pistil components and interactions leading to recognition, followed by acceptance or rejection. As mentioned earlier, following the compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grains, if compatible, will move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary. You would recall that in some plant, pollen grains are shed at two-cell condition, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms two male gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in the three-cell stage, pollen tubes carry two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. All these events from the pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. All the events from pollen deposition on the stigma until pollen tube enters the ovule are together referred to as pollen pistil interactions. All the events from pollen deposition on the stigma until pollen tubes enter the ovule together referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help A plant breeder in manipulating the pollen-pistil interaction, even in incompatible pollination, to get desired hybrids. So basically, it told that here it is mentioned about the two cell to three cell transition of pollen grain. Here, about how pollen interacts with pistil, with stigma, and stigma decides if pollen is compatible or not. Mediated by chemicals, which are only found out, only some of which are found out right now. And this part is about manipulation of pollen pistil interaction getting desired hybrid. Yes. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, corotalaria, balsam, and vinca on the vinca rosea on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of a microscope. You are likely to see pollen tubes coming out of the pollen grain. An experiment you should do in second. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiment, it is important to make sure that one desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. Let's read it again. Breeder is interested in crossing different species and often different genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. So that much is getting the desired hybrid. This is observing pollen tube generation, pollen tube generation. This one is the introduction to artificial hybridization techniques. Okay. Which are the emasculation and bagging techniques. If the female parent bears bisexual flowers, Removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flowers attains receptivity, mature pollen grains are collected from anthers of the male parent, are dusted on the stigma, and the flowers are rebagged and the fruits allowed to develop. If the female parent produces unisexual flowers, yes, just crashed, I believe. Yes, if the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be converted with a bag of suitable size, generally made of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attains receptivity, mature pollen grains collected from the anther of the male parent are dusted on the stigma and the flowers are rebagged and the fruits allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and flower rebagged. This is a brief introduction of the emasculation and bagging technique. So the pollen from here on, the pollen pistil interaction with stigma happens through chemicals which are not yet completely identified, and if the interaction dialogue between stigma and pollen is deemed that the pollen is incompatible, the stigma will reject the pollen germination or discourage the pollen tube germination. This is the pollen pistil interaction, and normally we saw that 60% of the plants, angiospermic plants, release the dehiscence of the pollen. Dehiscence of the anther will happen at the two-cell stage itself of the pollen grain, and when this pollen at the two-cell stage lands on the stigma and is allowed to germinate the pollen tube, then the vegetative cell and the generative cell of the two-cell stage, the generative cell will undergo mitotic division to form two male gametes which enter the ovule. The pollen pistil interaction and start from the deposition of pollen on the stigma and end at the entering of the pollen tube into the ovule. If we know what chemicals and what the pollen pistil interaction brings, we can manipulate it by using various non-compatible pollen grains to get superior varieties of the commercial superior varieties. And we can study pollen tube germination by taking some examples of flowers such as pea, chickpea, Crotalaria, balsam and vinca on a glass side and add a 10% sugar solution to it, and about 15 to 30 minutes you can observe a pollen tube generation. And they introduced, for artificial hybridization, they introduced two techniques of emasculation and bagging techniques. Emasculation is the removal of the anther from a bisexual flower to avoid contamination, and bagging techniques is covering the flower by a polythene bag or a butter paper to avoid contamination of the stigma or the flower. And that's pretty much it for the emasculation and bagging techniques. And with the next topic being the fertilization events, which is double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. After entering one of the synergid, the pollen tube releases gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus. The main point here is, one of the main points I have seen given question is, when does the or where does the pollen tube release gametes? Answer: in cytoplasm of synergid. One of the male gametes into the cytoplasm of the synergid. One of the male gametes moves toward the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, the pen. As this involves a fusion of three haploid nuclei, it is termed as triple fusion, since two types of fusion, syngamy and triple fusion, take place in an embryo. The phenomenon is termed as double fertilization because two types of fusions of triple fusion between the central nuclei and the male gamete, which is termed as triple fusion, and syngamy between the egg cell and the male gamete. It is because of these two fusions, it is referred to as double fertilization, an event unique to the flowering plants. The central cell after triple fusion becomes the primary endosperm cell, and one point here you should remember is, according to NCERT, this event of double fertilization is unique to flowering plants, angiosperms. The central cell after triple fusion becomes the primary endosperm cell, PEC, and develops into the endosperm, while the zygote develops into an embryo. This is the degenerating cell after fertilization. After fertilization or double fertilization. Double fertilization. The cell, the embryo sac will degenerate. This is the degenerating synergids. A zygote will develop. The primary endosperm nucleus will develop, but antipodals and the synergids will degenerate. And this is the zygote with the primary endosperm nucleus. The zygote will continue to divide mitotically. This is the two-celled stage. This is the globular embryo. This is the heart-shaped embryo, where this part is called the suspensor, and this is the complete mature embryo, where these two become the cotyledons, where it is a dicot plant, a radical, and the plumule here. Completely developing into a seed. This is the fertilization event of the double fertilization. The stages will be the two-celled stage, four-celled, and as so on. And the next stage will be the globular stage. The third stage will be the heart-shaped embryo. The fourth will be the final mature phase or mature state. Fertilized embryo sac showing zygote and primary endosperm nucleus, the A1, this one, and stages in embryo development in dicot. This is for dicot. As we see two cotyledons here, it means that it is a dicot plant. Post-fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds, and ovary into fruit are collectively termed as post-fertilization events. And the categories inside the post-fertilization events are events of endosperm and embryo development, maturation of ovules into seed. The first one being development of endosperm and seed, endosperm and embryo. Second being maturation of ovules into seed. Into seed and ovary into fruit. Ovary into fruit. These two encompassing all these events comes under the post-fertilization structures and events. Post meaning after. Let's start with endosperm. Endosperm development precedes embryo development. Before embryo development, endosperm is developed. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and used for nutrition of developing embryo. A main point that might be asked is that a statement based question, endosperm development is before embryo development. Why this happens is because as this endosperm, the triploid endosperm, continues to divide, it will form a endosperm tissue which is highly nutritive and will provide nutrition for the embryo development. As such, embryo development happens before the embryo development. The endosperm development happens before the embryo development. Why this happens is because after endosperm develops, it will give nourishment to the embryo development. That is why we learn endosperm first in the post fertilization events instead of the embryo or embryo structure. Nutrition of the developing embryo. In the most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm development. Such subsequent cell wall forms occurs and the endosperm becomes cellular. This free nuclear endosperm where we see is coconut water. The coconut water is nothing but the free nuclear endosperm this contains only the nucleus, whereas the kernel of the coconut is the it is the cellular endosperm. The coconut water is free nuclear endosperm. Occurs and endosperm becomes cellular. This free nuclear endosperm, if it establishes a cell wall, it will become cellular endosperm. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei. The surrounding white kernel is a cellular endosperm. Endosperm may either be completely consumed by the developing embryo. Example, in case of pea, groundnut, beans, such seeds are exalbumous seeds. Before the seed maturation, or it may be persist in the mature seed. If it persists, it's called albuminous seed. Castor and coconut, and be used up during seed generation. Split open some seeds of castor, peas, beans, groundnut, fruit of coconut and look for endosperm in each case. Find out that whether the endosperm is persistent in cereal, wheat, rice, and then The break took longer than expected. Now let's go back. So post-fertilization events, endosperm development precedes embryo development. Why? The primary endosperm cell divides and forms a triploid endosperm tissue. The cells of the tissue are filled with reserved food material and are used for nutrition for the developing embryo. In the most common type of endosperm development, the pen or the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclear endosperm. This stage of endosperm development is called free nuclear endosperm. Subsequently, later, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is a cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea groundnut beans. Pea groundnut beans are an example for exalbuminous seeds before seed maturation, or it may be present in the mature seed. Example is castor and coconut, and the use of this seed generation used up during seed germination. Split open some seeds. This is an activity. Split open some seeds of castor, bean, peas, or whatever you have availability of, and see if you find something similar to endosperm. If it is present in wheat, rice and maize. And this is the simple diagram of a typical dicot and monocot embryos. This one is a dicot embryo. These both are cotyledons. This is the plumule. We can call this as a shoot apex as well. This is the radicle, and this is the root cap. This region being below the cotyledons is called hypocotyle region. Next, let's go to the monocot embryo. In the monocot embryo, scutellum is nothing but a cotyledon. Cotyledon in monocots is called scutellum. This is a cotyledon as well, but it did not develop. As such, an epiblast is formed. This part is what we call an undeveloped cotyledon, which we also called as epiblast. And as for other, this part is the root cap, this is the coleorhiza. Coleoptile and coleorhiza can only be seen in monocots. This is the shoot apex here. This is the plumule region, radicle, root cap, coleorhiza and coleoptile. Now let's see the embryo. Next topic, embryo. And as for the before topic, endosperm, we saw that there were two types of endosperm, of one free nuclear as for coconut water, and two cellular as for the kernel in the coconut, the white color kernel. Next topic, embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. Because endosperm, we already read here in the endosperm development that endosperm development precedes the embryo development because the cells of the endosperm tissue are filled with reserve food materials and are used for nutrition for the developing embryo. As such, most zygotes divide only after some amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocots and dicots. Figure 1.13, this is the dicot embryo, this is the monocot embryo, depicts the stages of embryogeny in dicotyledonous embryo. We saw the globular shaped, heart shaped and mature embryo. That is what they are saying here. The zygote gives rise to proembryo and subsequently to globular, heart shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledon. This is the embryonal axis and two cotyledons. We see that this both are cotyledons. 1, 2 here, whereas we only see 1. The portion of embryonal axis above the level of cotyledon is called epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledon is a hypocotyle that terminates all its lower end of the radical or root tip. The root tip is covered by a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated toward one side of the embryonal axis. At its lower end, the embryonal axis has the radical and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, example wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of embryo and seed. You will see some development. Let's see it again. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocots and dicots. A dicotyledonous embryo, the zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryos. A typical dicotyledonous embryo consists of an embryonal axis above the level of cotyledons, the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the cotyledons is the hypocotyle. It ends at the lower end of the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons possess not only one cotyledon. In the grass family of cotyledon is called scutellum. It is situated towards one side, lateral side of the embryonal axis. At its lower end, the embryonic axis has a radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, and yes, that's activity. The next topic under and the third topic under post-fertilization and events, structures and events. It comes the seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of a seed coat, cotyledon, an embryo axis. The cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may be non-albuminous or exalbuminous. Non-albuminous seeds have no residual endosperm, as it is completely consumed during the embryo development. Example, pea and groundnut. Albuminous seed retain a part of endosperm, as it is not completely used up during the embryo development. Example, wheat, maize, barley, castor, etc. Occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also present. This persistent nucellus is what we call perisperm. Perisperm is not for endosperm, but remnants of nucellus is what we call perisperm. Nothing related to endosperm. Integument of ovules harden as tough productive seed coats. The micropyle remains as a small pore in the seed coat. Even in the seed, if we see, in seed there will be a scar, which is what we call hilum, which is the attachment site between ovule and the funicle, and there will be a small pore of the micropyle. Micropyle is not just a remnant but will also help in the germination process by absorbing water, etc. As the integuments of the inner and outer harden as the ovule becomes seed, the integuments will transform into testa and tegmen, which are the names. This facilitates entry of oxygen and water into seed during the seed coat. Micropyle remains as a small pore in the seed coat. This micropyle facilitates an entry of oxygen and water into the seed during germination. As the seed matures, if its water content is reduced and seed becomes relatively dry, 10 to 15% moisture by mass, the general metabolic activity of embryo slows down. The embryo may enter a state of inactivity called dormancy. Let me read again. As the seed matures, its water content is reduced and the seed become relatively dry. The percentage when we call the seed dry is if the moisture percentage is only 10 to 15% per mass, when the moisture content is only 10 to 15% per mass, this is when we call the seed has become dry. The general metabolic activity of embryo slows down. The embryo may enter a state of inactivity called dormancy. When such a critical state of water is reduced, the embryo metabolic activities will reduce and it will enter dormancy period. If favorable conditions are available, adequate moisture, oxygen and suitable temperature, they germinate as ovules. They germinate. As ovules mature into seed, the ovary develops into a fruit. That is, the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of ovary develops into the wall of the fruit called pericarp. The ovarian walls will develop into the fruit, and the wall is called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Classification of fruits and their dispersal mechanism we have studied in an earlier class. Is there any relation between number of ovules in an ovary and the number of seeds present in a fruit? It is. Ovules only will turn into seeds. As such, number of seeds and ovules will be same. Fertilized ovules will turn into seed. In most of the plants, by the time fruit develops from ovary, other floral parts degenerate and fall off. However, in few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits. True fruits are when ovary will develop, ripen into a fruit. This is what we call true fruits. True fruits are when ovary is the one that will mature and ripen to become the fruit. But in some special cases, in some fruits like apple, strawberry, cashew, the fruit, it will be something like this, where the cashew nut is embedded like this. This is a cashew fruit. This is the fruit part. In these fruits, the fruit doesn't develop from the ovary, but the fruit in the above cases develops from the thalamus, the base part of the flower. When fruit is developed from non-ovarian parts, not ovary, then that kind of fruit We call it as pseudocarp, false fruit. And eucarp or true fruit, if it's a true fruit, it should develop from ovary. In these examples, apple, strawberry, and cashew, the fruit will develop from the thalamus and not the ovary. The thalamus also contributes to the fruit formation. Such fruits are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruits are the result of fertilization, there are few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such an example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Normally, if you see a banana cross section, you see some spots in the banana. These spots are where the spots of the seed, spots of the ovules actually. They're not seeds, they are in a place where seeds should be there, because as banana is a natural seedless fruit. These spots, if you take a cross section of a banana, the spots you see in the center are where seeds, the spots of the seeds. And this banana is a natural parthenocarpic fruit. Parthenocarpy meaning the development of fruit without fertilization. Yes, and parthenocarpy can be induced artificially even in other fruits or other plants using special hormones, plant hormones, phytohormones. Let's continue. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperm. Firstly, since the reproductive process such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal to new habitat and help the species to colonize in another areas. As they have sufficient food reserves, young seedlings, before continuing, this point must be remembered. Seed formation is more dependable on water than pollination and fertilization. Pollination and fertilization are independent of water, whereas seed formation is dependable on water. I have seen this question asked numerous times, so remember this point. It's a statement based question. Pollination and fertilization do not require water, but seed formation does. Seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection of the young embryo. Being products of sexual reproduction, then generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Dehydration, the importance of dehydration and dormancy. I said that when the moisture content reaches 10 to 15% per mass, the seed embryo will reduce its metabolic activity and has a chance to undergo dormancy period, which will help in storage of the seeds for the next season planting in agriculture. You can't imagine agriculture in the absence of seeds, or in the presence of seeds which germinate straight away soon after formation and cannot be stored. How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seed we saw a concept of viability in pollen grains, where a pollen grain from cereals such as rice, wheat only lasts 30 minutes, whereas from the families of some special members from the families of Rosaceae, Leguminosae, Liliaceae, the viability of pollen lasts for months. The same concept is for seed viability as well. In a few species, the seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. The most common viability period as seen in large number of species is several years, with the least value being several months, the longest viability period being hundreds of years. Of viability. There are several records of very old yet viable seeds. The oldest is that of a lupine, Lupinus arcticus, excavated from the Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years of old viable seed is of the date palm Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. Examples being the first being the Lupinus arcticus, Lupinus arcticus, excavated in the Arctic tundra region, which had a viability of 10,000 years. and the second being the recent record of Phoenix dactylifera or the date palm seed, a record of 2000 years excavated from King Herod's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. If I had to answer these questions, how many eggs are present in an embryo sac depends on species. How many embryo sacs are present in an ovule? It depends on, there is a condition we will study, that is polyembryony here, where the nucellus will penetrate into the embryo sac and will form an extension of the embryos. There is also a condition where there will be numerous embryos, but the normal trend is one embryo. That is why we are studying it. And how many embryo sacs are present in an ovule? That is the answer, is how many ovules are present in an ovary. That depends on the species as well. And normally we saw that in the anemophilous flowers, we saw that because of the less chances of fertilization via the wind, the most of the wind pollinated flowers only had one ovule in the ovary. How many ovules are present in the ovary? It depends on the species, the type of pollination, the adaptation of the flowers. How many ovaries are present in a typical flower? In a monocarpellary condition, only one carpel will be there. In a multicarpellary condition, there will be many carpels and so on. So it depends on the species. How many flowers are present on a tree? That depends on the tree size, age, etc. Can you think of some plants in which fruits contain very large number of seeds? The orchid fruits are one such category, and each fruit contains 3000 or so tiny seeds. Similar is the case in fruits of some parasitic species such as orbanche and striga. Have you seen a tiny seed of ficus? How large is a tree of ficus developed from that tiny seed? How many billions of seed does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? So the main points here is orchid fruits, which contain very large number of seeds, thousands of tiny seeds, and similar in case of fruits of parasitic species of orbanche and striga, and a tiny seed of ficus. These are the main point. Orchids, striga, and orbanche, where orbanche and striga are parasitic plants, have humongous amounts of seeds in a single fruit. Let's go to the last topic. Apomixis and polyembryony. All those seeds are for a general apomixis is nothing but asexual reproduction mimicking sexual reproduction is called apomixis. Asexual reproduction mimicking sexual reproduction is called apomixis. Polyembryony is just a single ovule having more than one embryo. Let's continue to read. Although seeds in general are actually the one of the topics that come under apomixis is advantage of apomixis. The advantages of apomixis is again similar. If you remember, we saw advantages of cleistogamous flowers in autogamy, under autogamy. It will be similar. Here also you will get the assured seed set, because if asexual reproduction, it is not really true sexual reproduction, and there will not be a genetic change. As such, the seed set will be assured. It will be identical to the parent plant, and pretty much everything that is related to self reproduction. Let's just read for the products of fertilization. Seeds in general are the products of fertilization. A few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization is called parthenocarpy. Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of the apomictic seeds. In some aspects, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization, more often as many seen in citrus and mango varieties. Some of the nucellar cells surrounding the polyembryo sac start dividing, protrude into the embryo sac, and develop into embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Yes, as it is asexual reproduction, we can indeed call them clones. As for rereading this again, as it will be identical to the parental plant. Apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seed. One of the following is a diploid egg cell is formed without even reduction division. Normally, a egg cell is haploid, but here in some cases it might actually be formed itself is diploid. So, and it without fertilization itself, it will develop into an embryo sac, develop into an embryo. Hybrid varieties of several of our foods and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. This is a problem, remember this. One problem with the hybrid is hybrid seeds have to be produced every year. We can't use the same seed because it might have undergone a mutation, or it might have undergone something else as such. And because the farmer needs to purchase these seeds, these special hybrid seeds, every time, it will be a headache. But what if, because of advantage of apomixis is, with apomixis, the seeds produced will be same as the identical to the parent plant. The seeds produced will be same as the hybrid the farmer already purchased. So with this, the farmer need not to purchase the hybrid seeds once again. Purchase hybrid seeds once again and use the seeds from his harvest. This will help a lot of poor farmers. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. The production of hybrid seeds is costly, and hence the cost of hybrid seeds become too expensive for the farmers. If these hybrids are made into apomictics, there is no segregation of character in the hybrid progeny, then the farmers can keep on using the hybrid seeds to raise new crop every year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. The summary: flowers are the seat of sexual reproduction in angiosperms. In the flower, androecium consisting of stamen represents the male reproductive organ, and gynoecium consisting of the pistil represents the female reproductive organ. A typical anther is bilobed, dithecous, and tetrasporangiate. Pollen grains develop inside the microsporangia. Four wall layers, the epidermis, endothecium, middle layers, and the tapetum surround the microsporangium. Cells of the sporogenous tissue, lying in the center of the microsporangium, undergo meiosis, microsporogenesis, to form tetrads of the microspores. Individual microspores mature into the pollen grains. Pollen grains represent the male gametophyte generation. The pollen grains have a two-layered wall, the outer exine and the inner intine. The exine is made up of sporopollen and has germ pores. Pollen grains may have two cells, a vegetative cell and a generative cell, or three cells, a vegetative cell and two male gametes. At the time of shedding, the pistil has three parts: the stigma, style, and ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The serial tissue is the nucellus, in which the archisporium differentiates. A new word. The archisporium differentiates. The central tissue is the nucellus, in which the archisporium differentiate. A cell of the archisporium, the megaspore and mother cell, divides meiotically, and one of the megaspores formed the embryo sac. The female gametophyte, the mature embryo sac, is 7-celled and 8-nucleate. At the micropylar end is the egg apparatus, consisting of two synergids and an egg cell. At the chalazal end are three antipodals. At the center is a large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grains from anther to stigma. Pollinating agents are either abiotic, wind and water, or biotic, animals. Pollen pistil interaction involves all events from landing of pollen grains on stigma until the pollen tube enters the embryo sac, when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style, enter the ovules, and finally discharges two male gametes into one of the synergids. Angiosperm exhibits double fertilization because two fusion events occur in each embryo sac, namely syngamia and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. In the primary endosperm cell, the zygote develops into the embryo, and primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes the development of the embryo. This should be remembered again. The developing embryo passes through different stages such as the proembryo, globular embryo, heart-shaped embryo. Before maturation, mature dicotyledonous embryos has two cotyledons and an embryonal axis with epicotyl and hypocotyl regions. Embryos of monocotyledons have a single cotyledon. After fertilization, ovaries develop into fruit and ovules develop into seed. A phenomenon called apomixis is found in some angiosperms, particularly in grasses, results in the formation of seeds without fertilization. Apomictics have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony. With this, class 12, chapter 1, sexual reproduction in flowering plants, is completed. Based on NCERT. Congratulations. See you in next class.